Okay, today we are going to complete the last lesson, which is section 6 in chapter 9, genetic screening and gene therapy. As we said yesterday, that genetics provide a basis for new medical treatment. Okay, who can remind us with the genetic screening? Does genetic screening mean? What does genetic screening mean? Yusra.
Screening is the process of testing DNA of determine a person's risk of having or passing on a genetic disorder.
Yes, هي it helps in. Again, Yusra, can you repeat it again, please?
Genetic screening is the process of testing DNA to
Mhm.
determine a person's risk of having or passing on a genetic disorder.
يعني هو techniques or different techniques to test the DNA. Excellent, Yusra. And this also
Thank
helps determine that if any patient is at risk for certain disorder. So how this genetic screening would be helpful? Okay, if I make a genetic screening and I test the DNA of a patient, how this will be helpful? How we can determine? Okay, if we determine certain disorder, Irene, how we can help in changing this disorder? How we can help it? Yes, yeah, Abdul Tadil,
نعم مس.
how we can help if we make genetic screening? Can this help? Okay, we detect
Okay,
certain risk.
yes, it can
Yeah.
because it detects genetic disorders such as Duchenne's, and it determines the risk that you're willing to take or not to take.
Yes.
And it's used to detect specific genes or proteins.
Mhm.
It detects some genes that have a risk of cancer, like, for example, the pedigree and family history.
Okay, bravo.
What
And
else?
after detecting or determine these risks, can we help? Can we use these information for treatment in some cases?
I think yes, yeah.
Yeah. Yes, because so it will be helpful because in some cases or in some disorders we can make a change. Not in all disorders, in some risks or in some disorders we can make change. Today we are going to learn how we can make change or we can have new medical treatment for these risks or for these disorders. Okay. Now, here, as we said yesterday, anyone could be a carrier for genetic disorder, and genetic screening detects genetic disorders. We could be carriers, and we don't show the symptoms of certain disorder. So, genetic screening will help us to detect genetic disorders. Can you give me some techniques for genetic screening or for testing DNA? أعطوني يلا I will choose مراد.
Okay, some methods for screening اللي
مهم.
هو أول واحدة اللي هو electrophoresis والبيدجري وال
pedigree
وال
برافو
البانت سكوير
electrophoresis. What else؟
البانت سكوير
البانت سكوير. بس أكثر إشي عندي البيدجري is used for counseling يعني عشان to take information about the history of the patient أو the couple. بيعطيك history by counseling زي استشارة يعني. Okay, by asking you question about the history of the family and the disorders that exist in certain family. And from this pedigree, they can predict the risk for the offsprings or for the children. Okay, and the other way by testing the DNA itself by using electrophoresis and also maybe by karyotyping. ممكن الkaryotyping help us. So they test the DNA in different methods to make a genetic screening, a whole genetic screening, and help them to detect genetic disorders. Thank you, Murad. Genetic screening is the process of testing DNA to determine a person's risk of having or passing disorders. and it often involves pedigree and family histories as well as DNA testing in different methods. And we saw here that as you compare these 4 samples, عندي هنا this is a certain gene, يعني هون this screening for DNA or testing DNA determine risk of having or passing certain genetic disorders, not all. We can pick certain genetic disorders and such as the genes related to increased risk of cancer or other disorders such as Duchenne, which is a muscular dystrophy. يعني هذا مرض بيصيب العضلات. يصير عندي muscular dystrophy where the patient will have this case, they cannot walk and stand up properly as the toddler is going on and on. Okay? Sometimes you didn't see the symptoms. يكون عندي أنا المريض doesn't have the symptoms. ولكن over time the patient will show muscular dystrophy and they will not be able to stand up or move properly. فهدول الgenes they can be detected, okay, and cause these disorders such as cancer and like this disorder which is Duchenne. And they compare here the bands of DNA through electrophoresis with 3 samples مع normal sample, how it could be. By comparing these bands, we can detect if they have the risk or not. Okay? Now, but after genetic screening, we can use genetic screening for certain genetic disorders to have some treatment with gene therapy. We can treat some disorders with something called gene therapy. كيف احنا بنعمل medical treatment with medicines for certain diseases؟ هون بنعمل احنا they can treat some disorders, genetic disorders by the gene itself. Gene therapy. عشان هيك بنسميها gene therapy in which the gene replaces defective or missing gene. يعني حدا عنده مثلا they detect بهذا الجينيتيك DNA electrophoresis جيل الكتروفوريسيس لقوا انه في عندي ديتكت في جين للكانسر، تمام، او ديتكت لجين في الكانسر كان بلاد كانسر، تمام. هذا البلاد كانسر they want to find some gene therapy for it. They could find for this certain cancer هذا بالذات لقوا له علاج بالجينات. كيف؟ هلا we'll see how. They replaces defective أو missing gene، يا بيكون عندي أنا جين فيه defection فيه غلط أو بيكون في جين miss. They replace it with new gene. الجين الجديد هذا which is a gene that replaces the defective gene and this will treat this disease. Okay? هلا how they can do this? First من الفكرة الأساسية أنا عندي patient في عنده defect cells فيهم defective gene زي اللي عنده blood cancer. Okay? هلا they know where is the defective gene، بيقدر يعرف وين الجين اللي فيه defection فيه خلل. الفكرة الأساسية إنه to get the new gene اللي هو the other gene to replace okay the defective gene in the correct cells of the patient. هلا بس يدخلوا هذا الجين على cells of the patient الجين become part of the cell DNA بتصير part من cells DNA. اوكي طيب عندي سؤال قبل ما أكمل. هلا can this method used for any type of cell in your opinion؟ شو رأيكم؟ بقدر أخذ أنا أي cell and insert أو replace gene with the missing gene or defective gene and it will be part of DNA that makes common sense for you.
مس
كيف بده يصير part؟ انتوا برأيكم كيف يصير هذا part من الدي ان اي تبع كل البادي؟ يس محمد.
نسيت اسم أخذنا عنهم درسناهم إحنا
اللي هم ايش؟
مش موجودين في البون مارو اللي هم الستيم
ايوه برافو عليك
stem
excellent thank you very much يا محمد excellent. هلا انا I can do this but not I cannot do this in all types of cells. يعني انا عندي سيل اصلا موجوده زي السكن سيل. طب ما هي السكن سيل already عندها دي ان اي. طب if I take one cell وحطيت فيها جين، حطيت فيها correct gene or insert new gene، طب this will change only the DNA in this cell صح؟ بس انا هذا مش هدفي، انا هدفي انه السيلز كلهم to have a new genetic او gene part بالدي ان اي تبعهم. فهذا it doesn't work for all the cells. I must choose the correct cell. في حالة الكانسر of the blood cancer، مين هو المسؤول to make the blood cells؟ مين بيعرف؟ Which part of your body is responsible to make new cells, new blood cells? مين؟ How they are made? Yes يا لوجين.
They are made in the bone marrow.
برافو. هلا احنا بنعرف انه البلاد سيلز او السيلز in general they have life cycle. Yes سديد او عبد الله. What do you I would like to add something?
No, teacher, I was just going to answer the same
Okay.
thing.
I want to tell you something. هلا احنا we know انه في عندي السيلز، في cells they are replaced every day, every 2 weeks, and في cells عندي life cycle تاعتهم بعده years. Okay? مثلا the skin cells they are replaced. دائما بصير عندي division and new cells will be produced will all the skin will die. صح؟ في عندي كمان other types of cells like the blood cells. The blood cells they rid the blood cells every 3 months. كل 3 أشهر تطلع عندي new blood cells. Will all the blood cells خلاص they will go out the body will get rid of it by the skin. طحال. Okay? فهدول red blood cells will be replaced with the new cells. حيطلع بدالهم new cells. How they are replaced by the bone marrow. البون marrow اللي هو نخاع العظم is the part of your body which is responsible to produce new cells. بيكون جواته stem cells that they are not differentiated لسه ما صارهم differentiation and they will differentiate to new cells. Okay? ف what they are going to do now to make this gene introduced to these cells بياخدوا هذا الجين okay to take the sample of bone marrow stem cells بياخدوا sample من البون marrow stem cells and infect them with a virus that has been genetically engineered with the new gene. Do you remember لما كنا ناخذ البكتيريا نحطها في البلانت تكون البكتيريا فيها genetically engineered يعني عملنا فيها adding للجينز بالبلازميد صح؟ صارت البكتيريا transgenic بكتيريا. نفس الشيء they can use the virus ياخذوا الدي ان اي تبعه يعملوا له genetic engineering يدخلوا الجين اللي بدهم اياه في الفيروس then they infect the stem cells with this virus. فهذا الفيروس اللي فيه النيو تي ان اي مع النيو جين it will infect the stem cells then the stem cells are put back in the patient's bone marrow عشان هدول stem cells they can divide they have the ability to divide and differentiate they divide and make more blood cells with the gene يصيروا يعملوا division يطلعوا blood cells فيهم هذا الجين. Okay? So, if we want to do this, we should choose the correct cell of the patient's body. Again, I will show this for you in this diagram to make it more clear. Again, هلا أنا we said we should choose the correct cell, okay, to change in the gene. نعمل gene therapy. هلا they detect what is the gene. عرفوا إيش الجين اللي فيه defection أو فيه defection or it's missing. They want to replace it. How they are going to replace it? In this case, they use viruses, not bacteria. بياخدوا الفيروس. They take a DNA from the virus. Look at the virus DNA here. شوفوا في عندك blue part وعندي red part. The blue part هو الأول gene. they inserted in this DNA a gene, the wanted gene, اللي هو شايفينه بالred color. They inserted to the DNA of the virus. They put it back. Okay. صار عندي هذا الفيروس genetically engineered. Genetically engineered شو يعني؟ يعني صار في عندي altering في الجين، عندي تغير بالجين، صار عندي إضافة. It has genes from other organisms. هذا الفيروس نسميه vector. It binds to the cell membrane. بياخذ السيل cells من البون مارو. It binds to the cell membrane. They let this virus to infect it. يفوت جواها. When it infected, okay, بصير عندي هذا part of the stem cells. والstem cells طبعا الstem cells DNA والstem cells will divide and new cells will have this new gene. شايفين؟ Vector is packaged in vesicle. The vesicle breaks down releasing vector. Then cells makes protein using new gene. يصيروا يستعملوا هذا الجين الجديد to make that which express a new protein. Okay, and this is one way of gene therapy. Is one way of gene therapy using adenovirus in treatment of some cancer diseases زي blood disease. لأنه the blood is made or cells made by the bone marrow. والbone marrow فيه stem cells. Did you get the idea, guys?
Yes.
Sorry, any question about this part?
مس لو ترجع على ستيب نمبر 3 ما فهمتها كثير. الفصل يعني بعد ما الفيكتورز
هذا الديتيلز انا ما بدي تعرف الديتيلز هاي ديتيلز عشان to show you no problem. I don't want you to know انه what's going inside, but the main idea يا سبيل okay to know is we should choose the correct cells تمام that we want to make modification in it, genetic modification in it. باخذ انا هدول السيلز الستيم cells. I will infect these cells with a virus. هذا الفيروس before I infect it with the cell I should change its genes. اعمل له genetic engineering. تمام؟ بدخل الجين اللي انا بدي اياه in the DNA of the virus. هذا الفيروس صار عنده this new gene. Then I get it back in the virus and after that I let the virus infect the stem cells. بحطوها على الممبرين بعدين alone the virus will introduce to the inside the cell. يصير عندي vesicle. الفيسيكل breaks down بعدين it really هذا طبعا الفيكتور اللي هو الفيروس will go inside and this DNA طبعا هذا الجين become part of the DNA of the stem cell. فيه new gene and this new gene makes new protein okay for the cell. ممكن يكون هذا gene therapy for certain diseases. ونفس
تمام.
ممكن تزبط في other stem cells. طبعا هذا الجين therapy doesn't work for all the cases. لسه they are studying making some experiments about it. They couldn't cure every single genetic disorder by this therapy till now. They are trying, but they could make some therapies for certain disorders. هلا كان عندي the first successful trial of gene therapy took place in 1990. هي أول واحدة نجحت في 1990. The treatment was used on 2 children with genetic autoimmune disorder. كان عندهم مشكلة في جهاز المناعة. Okay? An autoimmune disorder and the children are now adults leading normal life. هي كانت أول trial of gene therapy. Okay? Now several experimental techniques are used for gene therapy. ممكن يستعملوا several techniques. التكنيك اللي احنا حكينا عنها هلا هي تاعت الفيروس اللي هي genetically engineered virus used to infect a patient cell. هي أول طريقة ممكن يستعملوها. يجيبوا فيروس. هذا الفيروس genetically engineered. شو يعني genetically engineered? Who can tell me what is the meaning of genetically engineered? What is the meaning of genetically engineered guys? Virus. Who remembered? Yes, Murad. Yes, Murad.
It means that it's genetically manipulated or modified.
يعني عليه يعني modified، صار هو اسمه transgenic virus. ليش؟ لأنه الفيروس الأساسي الدي ان اي تبعه كان بلو، عملوا فيه تغيير. they insert a new gene بدهم إياه في الدي ان اي تبع الفيروس من other organisms، فصار الفيروس ما عنده الناتشرال أو النورمال دي ان اي، لا، صار في عندي دخيل عليه. هذا الدخيل غير في الجينات تبعته للفيروس، فصار اسمه genetically engineered, manipulated, changed, altered. كل هدول نفس المينينج. هذا genetically altered أو genetically engineered virus can be used in gene therapy. هي أول طريقة. ثاني طريقة، في عندي sometimes they insert gene to stimulate immune system to attack cancer cells. أحيانا بيعملوها بطريقة مختلفة، إنه بيحطوا جين، هذا الجين مسؤول عن إيش؟ stimulating immune system، يحفز الجهاز المناعي. أوكي؟ to attack cancer cells. في عندي another technique insert suicide genes بسموهم suicide genes into cancer cells. هدول activate a drug so that only cancer cells are killed. عرفتوا؟ يعني بدخلوا جين مسؤول to kill the cancer cells. هذا الجين شو وظيفته؟ إنه activate a drug، كأنه بيشتغل عمل drug. أوكي؟ so that only cancer cells are killed، بس تقتل الكانسر cells. فهدول هم ال 3 methods of genetics أو gene therapy. Any question till here, guys? Okay. Now, gene therapy has many technical challenges. شو ال challenges اللي ممكن تصير عندي؟ أول شيء inserting gene into correct cell. مش إشي سهل. It's not an easy thing. Okay? هذا is a challenge to insert the gene into correct cell. Controlling gene expression. Sometimes when they insert the gene, they should يعني هذا الجين it keeps working and express when it's not needed. هذا الجين it's not needed. هذا يصير problem. فلازم يعمل في عندي challenge إنه how to control the gene expression. Okay? And determining effect on other genes. Sometimes لما ندخل جين جديد ممكن هذا الجين affect other genes. ف it's not an easy thing. After inserting the new gene, في challenges you researchers أو scientists should keep in concern. شو هم هدول challenges؟ إنه لازم يكون الجين to make sure إنه دخل على inserted correct cells. وafter they insert ده لازم يعملوا controlling لهذا الجين expression، إنه صار عندي الجين expression uncontrolled بصير يزيد الكانسر عندي مش يقلله، صح؟ وalso كمان بيشوفوا كيف تأثيره على other genes. It's a very complicated process, guys. Okay, now we finished the lesson. Any question? Any question? If there is anything not clear? Okay, so before we answer the questions, نعمل quick revision. We said إنه we can use genetic screening by testing DNA with different methods to determine if person is at risk or having or passing some disorders. قلنا genetic screening involves pedigree, DNA testing. Okay? They detect the gene, defective gene, and to tell the rest. Sometimes they can make therapy for this disorder, or they can tell about the rest. يعني أحيانا يروحوا الكابل بيعملوا genetic screening وبيقولوا لهم إنه you have risk for this disease, but we cannot do anything. There is no gene therapy. فبصير هون الديسيشن صعب. Should we complete together or should we leave each other? تمام؟ Not all the time it's helpful يعني. Sometimes يعني it have some problems. But if the people think for the future and for the health of their children, they should make يعني to be wise. They will decide if they want to stay with each other or they want to leave each other. depends on the personality of the person. Okay, so it will be sometimes helpful, sometimes it will be يعني not. So they detect the gene and they sometimes can find therapy by the gene, which is called gene therapy, by replacing of the faulty genes or defective genes or missing genes with new genes. Okay, يعني فكرة الجين therapy he replaces defective or missing genes or adds new genes to treat a disease. As we said, this is not a treatment for the whole genetic disorder. Okay, اللي في idea قلنا بياخدوا they choose the correct cells like the stem cells and they infect these cells with the virus. هذا الفيروس لازم يكون معمول له genetic engineering اللي هو genetically engineering يعني altered DNA ندخل فيه الجين اللي بدنا اياه. Then the virus will infect the cell وبصير عندي هذا الجين part of the DNA of the cell. ف when the stem cells divide بصير يعملوا division مع ال new gene express the new gene. Okay. And we said this is the first successful trial. هذا just for you to know يعني إنه عندي حتى two هم twins كانوا two children فيهم genetic autoimmune disorder and they now after genetic gene therapy they become normal with no problem. قلنا هدول هم techniques that we can use and these are the challenges إنه when you make the gene therapy you should keep in your mind to insert the gene into correct cells to control the gene expression and determine effect on other genes. Okay, let's answer the question. How does genetic screening use both old and new methods of studying human genetics? Yes, who can answer this question? Use both old and new methods of studying human genetics. Can anyone answer the question, guys? Yes, Yusra.
Miss, I can.
Genetic screening uses pedigrees and family histories as well as DNA testing.
برافو عليك. يعني أنا عندي genetic screening use both and the new both the old and the new methods of studying human genetics زي ما قلنا هون عندي البيديجري family histories برضه DNA testing. Okay, question number 2. Briefly describe the goals and methods of gene therapy. يلا يا سديل.
Okay.
إيش
So gene
goals
therapy.
and methods of gene therapy?
Okay, so it's basically the replacement of a missing or of a defective gene into a person's genome to treat a disease. And there are methods like, for example, it was talking about genetically engineered virus used to infect a patient's cells.
Yes, هم three techniques that we talk about اللي هم تاعت genetically engineered viruses.
Yeah, we'll insert gene stimulate immune system to attack cancer cells. The third one, I'm not sure. ما لحقت لا أكتبه.
الثالث واحد بنسميه suicide gene. هذا that kills the cancer cells stimulate as a drug يعني هلا we will talk about this. هيهم. The goal of gene therapy is to replace faulty or missing genes. تمام؟ هاي هي الجول للجين ثيرابي. أنا عم بعمل replacement للfaulty gene أو missing gene. In some cases genetically engineered viruses هاي أول تكنيك يا سديد. أول تكنيك هو by using genetically engineered viruses inserted into patient's body to deliver the function genes. Another method is to insert genes into cancer cells that will render them vulnerable to specific drug. يعني يخلي هدول السيل to stimulate specific drugs and to kill the cancer cells. The third method يا سبيل هيها stimulate immune system to attack cancer cells.
لا هاي عرفت أول تنتين عرفتني.
ثالث واحدة بحطوا genes هدول الجينز activate a drug so that only cancer cells بخلي هذا الدرug only kill the cancer cells.
Why does it say insert suicide genes?
يعني what is the meaning of suicide?
From what I know, there's a meaning of like suicide, like
Yeah.
killing yourself.
Yes. عشان killing.
So you guys put a killing gene in someone's. Oh,
Yes.
okay.
The
I get it now,
cancer,
okay.
the cancer cells only. Okay.
Yeah, I got it. Okay.
Okay, question number 3. Compare and contrast how is gene therapy similar to and different from making a transgenic organism. How the gene therapy is similar to making transgenic organisms first. How they are similar and how they are different. Who would like to talk about the similarity? يلا يا نجين، وين نجين؟ مراد.
Okay, the similarities is that in both transgenic organisms and the gene therapy, they both involve the change of specific genes inside the cell.
Okay.
They both involve modifying the
Yes,
cells.
both of them involve inserting a new gene. ممتاز. بالجين ثيرابي بالترانسجينيك organisms انت عم تعمل inserting لنيو جين في الاورجانيزم. اوكي؟ طب شو الفرق بينهم؟
اوكي الفرق بينهم انه
شو يعني transgenic organism؟
transgenic is an organism with a gene from different
from more than one species. From more than one species or from more than one organism. تمام؟ طيب هذا بالنسبة للترانسجينيك organism والجين ثيرابي.
أما الجين ثيرابي بيكون من اللي هو يمكن same organism.
لا مش نفس الاورجانيزم، هم نفس الشيء. هلا انت لما تحكي عن جين ثيرابي you are treating certain disorder. شو عم تعمل لما تعمل جين ثيرابي؟ you are replacing the defective gene with normal gene in adult organism. تمام؟ إذا أنت عم تعمل replacing لا defective gene with normal gene في adult organism. The transgenic organism has genes from more than one species. لما تكون أنت عم transgenic organism زي الفيروس مثلا أو البكتيريا، هذا transgenic organism فيه جين من more than يعني from more than one species أو ممكن one or more species. مش شرط يكون عندي more than one، ممكن يكون عندي one or more species. فالفكرة هي بالمعنى إنه هم both of them inserting a new gene into an organism. بالحالتين أنت عم بتدخل new gene بالorganism، بس بالجين ثيرابي it replaces defective gene. تمام؟ Transgenic organism شو معناتها؟ إنه they have genes from more than one species. Is it clear now مراد؟
Yes. يعني هسه قبل ما نسوي جين ثيرابي لمثلا شخص، هذا بصير ترانسجينيك.
برافو. الفيروس اللي عندك هذا صار فيه تغيير بالجينات تبعته، هذا ترانسجينيك اورجانيزم. تمام؟ هلا الجين ثيرابي بدك تعمله انت لادلت اورجانيزم. واحد adult يعني it's grown up and he is adult. اوكي؟ فانت بتاخذ هذا الجين اللي هو الجين to make the treatment، تعمل له replacing. تحط replace gene therapy replaces a defective gene مع normal gene in an adult. هذا هو جين ثيرابي، عم بتعالج حالة انت. اما بالترانسجينيك اورجانيزم لا، في عندي جينات has genes from more than one species، عم بتدخل جينات from more than one species عشان تعمل تغير في تريال معين to make change in certain trial. اما الجين ثيرابي لا، انت عم تعمل replacing in adult to make therapy, to treat something. Clear يا مراد؟
clear مثلا.
في حدا عنده سؤال على هذا السؤال؟ تديل.
No, I don't.
Okay, last question now. Synthesize how are restriction enzymes and recombinant DNA important for gene therapy. I will read this answer. As we said إنه كيف restriction enzyme وال recombinant DNA important. طبعا عشان أنا to insert a new gene بدي restriction enzyme صح؟ عشان to make recombinant DNA اللي هو DNA اللي فيه gene from other organisms. Restriction enzymes cut DNA to insert a gene into virus. لما يصير عندي الفيروس هذا فيه نيو جين بصير اسمه بصير عنده recombinant DNA اللي هو DNA with other genes from other organisms. Okay? Any question? Okay, guys. So tomorrow, inshallah, we are going to start reviewing. I will see you tomorrow.